I, for one, enjoy these. I've never done this before, where like at the beginning of class, I throw this thing out here. These like, especially with like these super really easy little things like this that you can do in like 2 minutes. Just to do this little kind of low-key informational reinforcement kind of stuff. I'm maybe overestimating with the 2 minute thing. Do you like doing these kind of things?
Yeah.
I mean, there's value, there's purpose in this. It not only reinforces learning, but it reminds us that we actually know sort of what's happening a little, right? Because you don't know that you know unless you find out whether or not you know. And so if nothing else, you use this as a super low-key, low-stress way to know that you know. And through time, you both get smarter in bio and confidence starts to go up. The downside is that both can cause friction. Both can cause friction. You can be perfectly right and still suffer pain. And that's usually the resistance caused by lack of confidence in knowing that you know what you know. So it's the least stressful way that I can think of to reinforce that you actually know that you know. And so it's like, you can't hide when you write. You know, every bit of you will be exposed for either the genius or the fraud that you are when you try to write down an answer for something. It's like you can't hide from that, you know. And typically, sorry, Brooke, that's proportional to the number of pronouns that we have. It's like I could grade these based on pronouns. If you have three or fewer pronouns, you get an A, right? If you have 3 to 6 pronouns, it's a B. If you have nothing but pronouns, it does the thing which happens to the other thing, and it goes up. It's like, okay, cool, awesome. And I get that. When I say I get that, I mean, I don't mean I understand it. I do understand it, but it's like people submit things like that sometimes, and it's like there's no there. It's like, I love you, kid, but we got to get some specifics on this. So always pay attention more than anything else in your science classes to your use of pronouns. Whenever you put a pronoun, we know what pronouns are, right? So, for example, glucagon is released, detected by the pancreas, detected by the liver, and then it goes up. What is it?
Insulin. No, glucose. No.
So it's like, yeah, so it's like we think we know what we mean by the word it, and I'm not saying that it's wrong. I'm saying that if there's a pronoun that you put on something, you run the serious risk of me calling you on it and saying, what do you mean by it? Right? Because it's like, that's where ignorance hides. Ignorance hides in pronouns. I'm not, sorry.
You gotta get tired.
Yeah, it'll be okay. Ignorance hides in pronouns a lot of times. So it's like it's okay to put them, I get it, right? I use them myself, but make sure that you're being aware of what that pronoun actually means. What is the noun behind it, right? And if you don't know, so I was, you could say sugar or something. That I was thinking blood sugar, but if you put it, it's like it. What is the it? What is the it? So good, lovely. Where did we leave up last time? What did we end with? I think we ended with
The cellulose.
cellulose. I think we put a pin in sugars. I think we're sugared up. I think we've been sufficiently unsweetened to move on to the next of our macro molecules. Would you agree? Enough sugar for one day?
Correct.
Perfect. Pour your sugar on me. What is that? Def Leppard from the 80s, I think. Sleepy time. It was a loud yawn coming out of them. You have seen a molecular structure like this when you were drawing Which of our macromolecules?
Tree.
When we were drawing lipids specifically, triglycer. It makes sense now, doesn't it?
Yeah.
Right? Perfect. What kind of glyceride? Not a monoglyceride, not a diglyceride, a triglycer. I was going to say I, but 6 1 half doesn't mean the other. Triglycerides, awesome. Perfect. This by itself is delightful. This is not the last time we will see something like this. And I'll talk more about that when we get into respiration in a couple of weeks. This glycerol looks like half of a sugar, right? It looks like it's a triose sugar. C6H12O, or it's like C3H8O3. I don't think you can't really circularize this because it would be too tight and it would interfere with itself. But this is essentially a triose sugar. So it's like we're talking about lipids and we started with a
Sugar.
Carbohydrate, which is weird, right? This will come back again in a couple of weeks. Functional group? What kind of functional group is this? Number of functional groups?
No.
I know what Tuesday's little quiz is going to be. Carb? Ox.
Carboxyl?
It's a carboxyl group. Yeah, it's a carboxyl group. Oh, it's a carboxyl group. Yes?
Yes.
So it's like we need to do some reinforced learning on our function. When I draw a squiggly line like this, that means like, and more of the same. Typical, just so for an FYI, this we would call a fatty acid. This is a fatty acid. What makes it an acid? You put these in some water and you shake it. That is typically how we measure acids. Let's make this an acid.
pH level smaller than 7.
Great. That's the numerical equivalent of what this would do in the solution if we did it. What makes this an acid?
Hydrogen ions.
What if sentences have nouns and verbs?
All of the hydrogen.
Not all of them. One. Only one comes off. Which one? Brooke, which one comes off? Which one? Yes, the one in the carboxyl group. It's almost always the one in the carboxyl group comes off. Bam, fist bump on the far. So it's a fatty acid. What makes it an acid? That hydrogen will pop off in solution times and it'll ionize, into a negative. But when we draw it, typically as a fatty acid, we put it on there. Why so? Why so? I'm going to draw another one. For these, I'm just going to go like that, if that's okay with you. Much like we may have done in our drawings that we submitted. It's like, it's just like a long hydrocarbon chain. I'm just going to kind of do this for efficiency. And a bunch of carbons on down. Cool. So right now I have 1 glycerol and 3 fatty acids. Is there some way that I can attach these fatty acids onto this glycerol? How so?
Backbone.
Yes, you do. You do know.
That,
The backbone.
one of the H's on the end combines with the. Yeah, it combines with the hydroxyl. Yeah,
What do we call that?
bonds.
The reaction is called?
Hydrolysis?
Um,
Yeah, hydrolysis.
It's not hydrolysis.
I don't understand break.
It's the other one.
Oh.
Hydration.
Dehydration. How many times?
3.
How many water molecules are going to come out?
3.
3.
And so, it's going to shuffle out. There's the oxygen, the carbon. So you have this oxygen that's left over that links the glycerol to each of the fatty acids. You have still has double bonded oxygen still hanging out over to the side. Everybody valence electron states are full and complete. Super stable molecule. This is now a triglyceride.
What's that word?
Which one?
The right one?
This one?
Yeah,
It's triglyceride, but I got tired and so I stopped writing.
the same thing.
Yeah, triglyceride. Yeah, triglyceride. So it's like 1 glycerol and 3 fatty acids will react together to give you 3 molecules of water through the reaction called dehydration, and your result will be a triglyceride. So this, like I was saying before, this is that metabolic water that I was talking about. Perfectly good water molecules. You can use them for all kinds of hydrolysis later on somewhere else. Ricky Brooke?
So when fatty acids attach to the glycerol, and all like the electrons and neutrons are good and everything, it becomes a triglyceride, and then that's when you're dehydrated.
Yeah, you dehydrated these 3 fatty acids onto this
With the-
glycerol.
Okay.
If we wanted to get these back, so, and this is where we're gonna, oh, cool. We kind of put this stuff together in long-term storage so we can get the energy out when we need it later. Where can we stash this once we build it?
What's that?
Right there. Right there. So I have mine stashed under my skin in the midsection, right below the Nova logo. If I want to get the energy back out of this, the first thing that I need to do is break this thing apart, correct? How would I do that? What's the first thing I would do to break this apart? Say it.
Hydrolysis.
Yeah, we're just gonna take the water, shove it back in, put the reassemble it together, right, and break them apart. And then we're gonna go back from having a triglyceride to a triose sugar. Oh, I can't help myself. I have to say it just because it's so joyful and gleeful for me to say. a triglyceride, a trio sugar that you can bang straight through the Krebs cycle and aerobic respiration. We'll get to that in a few weeks. But you have the 16 or 18 carbon long fatty acid. You have to break this apart as well to get the calories out. More on this in a couple of weeks. You'll start cleaving it into 2 carbon chunks called acetyl groups that you can then blang into the mitochondria straight away to get some ATP out as well. So you'll break this whole thing apart. Right? And so why go through the bother of building triglycerides from extra energy that we have around? That's it's high density energy storage. It takes up less space per calorie than carbohydrates do. You don't want your blood to turn into pancake syrup. Calories are sometimes hard to find back in human evolution. So anytime you get an excess of carbohydrates in a sustained way, it's not a bad idea to store them as high density storage somewhere else. And so you build these things out of it. And then if you put it right under the skin, you can then get the added benefit of insulation, impact resistance, all that other good stuff. So again, these fats, it's not just energy storage. They have these other properties you can monopolize on by where they're deposited in the body, right under the skin, which is kind of cool. And so in order to get to this part where you start breaking this stuff apart, to use that energy storage, you have to exhaust yourself of the glycogen in the blood sugar before you get there. That's the conversation we were having about keto and slow carb and all that kind of stuff on Tuesday. But you can do it, but it takes a little bit to get there. Questions, thoughts, comments? Wonderful. Should I take a picture of this? Okay. I'm going to try to salvage as much of this diagram as I can. If I take one of these 3 fatty acids off and attach a functional group that you have not yet been made aware of, that you'll want to know for Tuesday, you can make something else. So I will now show you the wackiest functional group that there is. When I say wacky, it's wacky for about 1 million reasons. It's called a phosphate group. What a weird group. Look at all these negative ions coming off of it.
DNA, right?
What?
It's the one in the DNA.
Yes. Stay tuned. What's that, Sylvie? All of a sudden you're synthesizing information in a fan. Yeah, eventually we'll use this phosphate group to link nucleotides together in a DNA chain, right?
It's like
Right.
like
And ATP, these are the P's in ATP, 3 phosphate groups. Okay, now, if we want to really flex do you remember valence electrons and octet rule and all that? What's wrong with this molecule? It's not in the oxygens. What is this? What's wrong with this? How many covalent bonds should we be able to make?
3,
3. How many do we have?
4, 5.
5. It has no regard for the rules of covalent bonding. Books have been written about phosphorus because it is so weird. It's like, it doesn't obey the octet rule. It's like, I only have 5 valence electrons, so I should be able to make 3 covalent bonds. No, I'm making 5. And if you don't like it, well, you know what? I'm still gonna do it. What are you gonna do? What are you gonna do? Right? It just violates all sorts of periodic table rules. Phosphorus. There's like books are written on phosphorus just because it's so freakishly strange. So freakishly strange. And fundamentally required for life on Earth for reasons that you just mentioned. It's like you literally attach nucleotides together to make DNA chains with this phosphate group. Without phosphorus, life on Earth would not exist. This sort of thought. It's like one of the few things that unifies life on Earth, like in total, is the use of phosphorus as a critical element within it. We link nucleotides together with it. We make phospholipids that we make cell membranes out of with it. We make ATP out of it. Some have speculated that the total amount of life on Earth that you can have on the surface of the Earth at any time is directly related to the amount of molecular phosphorus that exists on the surface of the Earth. It's like you can only make, it's like the limiting resource that determines how much life there could be, like how many DNA chains there could be on the surface at any time. To the zero-sum extent that if you want more life somewhere, if you want to add to the amount of life on Earth, or you want to have more life on Earth, in order to get that, you're going to have to take some away so you can free up more phosphorus to make more DNA chains. So it's like we're at the zero-sum state. To get new life, you have to get rid of the old life. Thankfully, everybody dies, but you need to liberate phosphorus in order to get more of it, which is well. Brooke, question?
Can it exist on its own, or does it have to?
What?
Can it exist on its own, or does it have
Like
to?
molecular phosphorus? Just like a pea floating in space?
Yeah, basically.
I've never
Okay.
heard of it, seen it. Very few things actually do
Okay,
exist
so
in
it
a
has
pure
to be
molecular
attached to
state.
something else, though.
If it's not, it will be very, very quickly. But I mean, it's pretty electronegative. I mean, it's up in that upper right. It's gonna, as soon as there's free phosphorus there, it's gonna grab onto something and make something out of it. But what a weird functional group. All these oxygens out there, all of which are extremely electronegative, right?
Yes.
Yes, cool. This molecule we would call a phospholipid. So we made a phospholipid by taking a triglyceride, removing a fatty acid, and substituting in a phosphate functional group. Cool. So if you know how to draw a triglyceride, you know how to draw a phospholipid because you just pull you pull that last fatty acid off and you put it in the phosphate group. In order to do that, you have to know how to draw a phosphate group. If you follow the rules of the periodic table, you're going to get it wrong because you're not going to have as many oxygens as you need. Phosphorus is weird. You can do 5 covalent bonds. It's a lot of oxygen kind of wrapped around this thing. It's a strange molecule. It's a strange one. But we use it all over the place in living systems, which is kind of wild. Cell membranes. We'll talk more about cell membranes in a week or two when we talk about cells and osmosis and how membranes work and things like that. Long story short, having this phosphorus molecule up here has real serious consequences for what this molecule does. It behaves in completely different ways now that it has this phosphorus group. I'm going to erase this and draw it smaller. Is that okay? Go ahead.
Okay. So the third C, right?
Yeah.
So that's when we go down and it makes it the phosphate.
Yeah, every time I think I've only ever seen it drawn where the phosphate group is one on the end. It's never one in the middle.
So we still draw those. We still have those.
These two?
Mhm.
Yeah. They are a critical part of it. They're a critical part of it. We used to draw every electron in things, and now we just kind of shorthand it by drawing the letter and things like that. Phosphate groups are usually a P with a circle around it, like that. So this indicates not phosphorus as an element, but the phosphate functional group as a whole thing. You with me? Clear? Good, perfect. And it has this big, huge negative charge hanging off of it, as you recall. Good? Which means the top half of this molecule is now polar. The bottom half is now nonpolar. The top half can dissolve in water. The bottom half is now cannot dissolve in water. We say these molecules are amphipathic. Amphipathic molecules are molecules where half of them are polar and half of them are nonpolar. And we did this here by adding this functional group to the top.
Yay. I was wondering.
Oh, Van, I watched a lot of Wheel of Fortune. Was going to, oh, Van. Perfect. Questions, thoughts? When we draw these as cell membranes, when we draw these throughout, we will start drawing them as a little circle with two little lines that come out of the bottom. This is shorthand for this whole molecule over here. Sound good? And we do that because when we draw cell membranes, let me go back. So if you have a bunch of these phospholipids and you put them in a mason jar with some water and you start to shake them up a little bit, you get this arrangement where it's like the water can only be in contact with the part of the molecule that has a charge on it for polarity charge reasons. And all the fatty acid tails that hang down have to be associated with each other, with none of them in contact with the water. And there are two ways that this molecule can arrange itself so that it happens. One way is they'll form little structures like that. So all the fatty acid tails in the solution are pointed inward, and on the outside of it are all of the phospholipid heads. You get that? Right, so this is all a bunch of water kind of out here. So sometimes, oftentimes, they will arrange themselves this way. All the heads are in contact with water. None of the tails are in contact with water. Like dissolves like, right? Smile and nod. Perfect. The other way is like this. They will form a big hollow sphere with the outer layer is the phospholipid with the charged head pointing up, and the inner layer is the phospholipid with the charged head pointing down, and the tails, the fatty acid tails, are still in contact with each other. So you have like water up here, water down here, but all the tails are still in contact with each other in a nonpolar way. So you get like polar side, you get a non-polar region, and then you get another polar side. And it forms a big sphere. So the inner and outer surface of the sphere are polar, but the middle part is nonpolar. Does this make sense?
Are the tails always going to be touching? Like the non-polars always?
They're close enough to count.
Okay.
Close enough. They're always going to associate with each other. They're not going to flip this way.
Yeah.
They can move laterally back and forth, but they cannot flip. They're not going to flip this way. They're not going to flip this way, and they're not going to flip over as an individual. When they're arranged like this, and we put lifey stuff on the inside, we call that a cell membrane. This is how every one of the cells in your body is organized, as a phospholipid bilayer, tails pointing in and the charged heads pointing out. Why is this a great cell membrane? It's great because all the stuff that has a charge on it, right, sugar molecules, salts, all the kind of stuff that you have, cannot pass through that membrane because all the charged stuff on the inside cannot make it through the fatty acid nonpolar tails because it's insoluble. Oh.
Is it the same one in the mitochondria?
Mitochondria has two of these. It's double membrane.
Can you repeat what you just said?
Yeah, because it was like a big one.
Mhm.
It's like, no, that's why we take Kinbio, you know? All the charged stuff that we have, sugars, the salts, all this other kind of stuff that has any sort of charge, either because it's ionized or because it's polar covalently bonded, that's on the inside of the cell, cannot diffuse out to the outside of the cell through this membrane because the charged stuff is repelled by the nonpolar fatty regions of this. Like dissolves like, right? So like water, we say water is the universal solvent because most stuff in the universe is charged. The inside stuff cannot diffuse out. The outside stuff cannot diffuse in. So they make really, really, really good cell membranes because they keep the inside in and the outside out. They're still able to associate with water because the outer surface is charged, but the nonpolar tails on the inside prevent things from crossing back and forth. Go ahead.
What does the small flower mean? I didn't catch it.
This thing,
Yes.
this does look like a little flower, doesn't it? It's one of the two ways that phospholipids, when they're in a solution, can organize. These are going to be really, really small, right? But it's one of the arrangements where the charged heads can be in contact with water, but the tails will be in contact with themselves. So this is one arrangement that you can get. The second arrangement is this bigger, complicated things. This is too small to hold life, and you couldn't hold life on it in the inside because you don't have an inner surface where you can put stuff into. So it's like just a little fat sphere. It's like a little fat ball, right? Like tiny, tiny, tiny. It happens, it's thermodynamically stable. The charges work out. You'll get some of these in solution. These, however, a lot bigger. You use these as barriers that you make cell membranes out of. Take your finger right now and touch a cell membrane. Touch one. It's like all these cell membranes, phospholipid biomarkers. Raise your hand if you have phosphorus. Through and through. Vanessa, do you have a question?
Oh, okay, so the thing that looks like a cloud, that looks like a peach.
Whoa, start over. It's like,
So
I heard syllables, but that was it.
Huh?
I heard syllables, but that was it. Go ahead.
the drawing that looks like it's a cloud with rain.
Like it's this
A
one?
cloud, no,
This one?
right.
Okay.
So the top portion is polar,
This is polar.
and that's not.
This is polar. This is nonpolar.
Oh, the middle.
The middle is nonpolar.
Okay, so the emptiness is not, or the.
It's like the part in between.
Ah, the emptiness. I love that. So like this part in this region is nonpolar. So it's like polar, nonpolar, nonpolar, polar.
Okay, that's
Right.
Okay,
Right.
thanks.
Go for it.
So there will be H2O life connecting from both sides, right?
Yeah, there's water in here. There's water out here. You know, here's my chalk. Is class over? What happened to my chalk? But know what to do with my chalk?
Check your pocket.
Like,
Is it the one all the way over there?
Hey, there's.
it might be because like I was all the way over there. I don't think.
No, I think that's the crappy chalk.
Yeah, this is a crack chalk.
I think your chalk wants less
Exactly. Did you tuck it behind your ear?
time. You're holding.
No, this is the crappy chalk.
Oh, why'd you have more?
I've done that before. I've done that before. It's like, I begrudgingly might have to use my, ooh. Because you have to find one with the logo on it, right? Gucci chalk. So if I actually drew an actual one of these phospholipid bilayers, look how beautiful it erases though. It leaves them. Whoop. Yeah. It's like, it's obnoxious, but it helps me draw it at least once just to, you know, just to see. Phospholipid bilayer, which we use to make some membranes. Does that make sense? So here, you actually have both of them. It's a bad drawing. It is a bad drawing, but you get the point.
It looks like a germ.
It does, right?
Like sunflower.
So this is polar on the inside,
Polar.
polar on the outside.
Would it get any life in the middle in general?
Every cell in your body has life in the middle in general. So that's where life happens inside of this.
And
Structure.
the outside, because the outside's polar too?
In here is life, right?
And then-
Outside of this is environment.
Oh, so this. Okay.
Everything inside of this you call life, everything outside of that you call environment.
So where the,
You have the reactions happening inside of this thing.
I know, so like in our cell.
The outside is environment, so where's the little fatty acids?
It was these,
Yeah.
but we converted them into phospholipids. This thing in an actual cell is going to be a gazillion of these. Like, it's going to be do this like the size of this room, okay? And that would be more appropriate to scale. I didn't want to sit here and draw fatty acids all damn day though, so it's like, I made an ugly one. But it's sort of this is larger than life size. Your cells are not actually this big, correct? But this is like depicted, sort of. But it's the whole circle instead of just like part of it with the rest of it. It's technically a sphere, right? Three-dimensional sphere. Go for it.
Is there any way for like something like that to like get bigger, or like swell up?
Yeah,
What is that?
that happens when you eat. That happens when you drink a huge amount of fresh water,
Beach.
right? So if you have like a whole lot of life happening in here, you got a lot of sugar, you got a lot of salt, you got a lot of molecules that have charges on them, and you have a whole lot of like drink gallons and gallons and gallons of fresh water, right? Water will be under high concentration here relative to this one, because this has more solutes inside of it. The water will flow in and the cell will swell up in a process called osmosis. More about that in a couple of weeks. Don't do it because it'll kill you. Because some of the first cells that swell up are the ones in your brainstem. And will block neural transmission between your brain and your spinal column, and you'll stop breathing.
Is there any way to permanently?
What's your objective? Let me ask you a different question. What makes you ask?
I don't know, I think my first thought was, I wonder if the bigger you get, the bigger your cells get, and I was like, no way.
Yeah, can be, right? The other way is to, every day go to Olive Garden and get the never ending possible.
Please,
'Cause,
no.
I mean, when you get,
I work there.
I mean, when you like, when weight goes up or down, when weight in longer term is gained or lost, short term, it's usually water. Long term, it's usually triglyceride fats. When you deposit fat, triglycerides in your body, because you have an excess of calories, you're not adding more cells. You're taking the fat storage cells and making them larger or smaller. Okay? Yeah, right. The question you got to the point was, what are you thinking? What are you trying to do?
There's a lot.
Well, you're going to go home with this information and you're going to.
Probably tells my brother about this.
Sweet. Does all this make sense? Go for it. What's
I
up?
was trying to go a week without hearing about Olive Garden. You ruined that for
Sorry.
me.
The last thing I want is for people to start like, no, I'm really in the mood for some Olive Garden. I don't know why. Subliminal. This lecture brought to you by Olive Garden. Questions about this?
So that small sunflower one, that's just a different arrangement, right?
Correct.
And then
Correct.
the one next to it is the same thing as the one you just.
Correct.
Just.
Correct.
uh,
Correct.
Okay.
Correct. It's like, this works too, but it doesn't do anything interesting, right? It's a stable arrangement that shows that the tail's associated with the tails and the head's associated with the environment. This is another way to do that, but this one is far more useful because it's not size limited like this one is. And those, therefore, are the ones you use to make cell membranes. If you pack life in there and call it yourself. Cool. Question. Go for it.
Well, the only thing that can get into that is fresh water.
Oh my God. This makes me so happy because you all are desperately attempting to take this information, synthesize it, and move on to the stuff in the next chapter, which I think is the bomb.com. You are starting to anticipate what comes next based on your understanding of what comes now.
Yes.
I will say, what an awesome question. What an awesome question, because where is oxygen? Where is it? Point to it. It's like right here. How do we get it? Where's it come from?
Trees.
Trees, photosynthesis, right? Where do we need the oxygen?
No.
We need it here. So how do we get oxygen from out here into this thing? Oxygen is polar, nonpolar. Bonded to itself. Nonpolar. So does oxygen being nonpolar have a problem getting into this? No, it can make it through the nonpolar tails. Nonpolar stuff can go back and forth as long as it's small. Right? Oxygen can diffuse in. All it needs is a gradient. As long as photosynthesis keeps happening, the oxygen can move in as long as our circulatory system is doing a good job of distribution. CO2, polar, nonpolar.
Polar.
It's nonpolar. It's a strange one. It looks polar, but it's like, so you'd think the electrons would be like way out here and way out here. They're not. They're pretty much dead center because this oxygen actually has so much of a pull that it pulls on these. This one. Where are you making CO2? You're making CO2 here. It's nonpolar. You're always going to have a higher CO2 concentration inside the cell. So what does it do?
That's it.
Diffuses out. It's nonpolar, can go through the tails. Free pass. What uses CO2 in the environment? Plants, photosynthesis. So it's like you're taking the waste product of plants, breathing it in, and through that metabolic process, producing CO2, which you then blow out and give back to the plants. That's that circle of life shit they were talking about in The Lion King right there. Because the actual relationship is not between animals and plants. The actual relationship is between mitochondria and chloroplasts, which are bacterial. Go for it.
So how does like oxygen?
Say again.
How does oxygen and carbon dioxide get through like the polar heads into like the?
They're in solution. Yeah, oxygen also has this weird ability to dissolve in solution. So does CO2. Like carbonic acid. So it's like it's a nonpolar molecule, but it still is so small that it can hang out in solution. Carbon dioxide, when it does so, we refer to it as carbonic acid, right? And so all of a sudden we start talking about body pH and things like that, and how much carbon dioxide is dissolved in your system. Oxygen is the same way. You can dissolve far less oxygen in water than you can have in an environment, but it's enough. So it's like, why do you use oxygen as a respiratory thing? Well, it's small and nonpolar. It can pass through the membrane just fine, and it's highly electronegative. We can do neat stuff with it. Waste product of CO2, awesome, small, nonpolar. It just diffuses out. All we have to do to get rid of it is rely on our circulatory system to distribute to our lungs, where we can just then blow it out, which is kind of cool.
Every time we take a breath, does it go through and out?
You have some other bio2 related complexities.
Yeah.
Usually when you're breathing in and out, you're not going, the oxygen makes a couple of stops along the way. I do this in Bio 102 where I do that? It is Bio 102, where the question is like, how many membranes does oxygen have to pass through to go from the environment to an actual inside of a cell? It's like it's got to go through lung epithelia, then it has to go into the capillary, then it has to go to the place where it needs to be, then it has to go out of the capillary, into the tissue, through the tissue. It's like seven, right? So nothing is a straight shot. And usually when you're breathing in, you're not doing straight, directly breathe in to the tissue itself, with the exception of lung tissues. Within your muscles, to get into the nitty gritty, within your muscles, you have a little bit of a buffer of stored oxygen. In your blood, you use a protein called hemoglobin to carry oxygen around. That usually does not deliver it directly to the tissue that needs oxygen. It delivers it to another molecule called myoglobin, which is in place in your muscles, and it stores oxygen and what makes muscle red, right? Iron containing acceptor. And it's like the oxygen you're really using when you're moving around is the myoglobin oxygen, which is usually released in response to pH. When you start to produce a lot more CO2, the local tissues become acidic because you're releasing that CO2 and making carbonic acid out of it, which makes the pH go down. And myoglobin, in response to lowering pH, releases oxygen because it uses the pH drop from the carbonic acid being produced as a trigger to release more oxygen in that place. So using that mechanism, you can selectively release oxygen from myoglobin in different parts of your body based on what muscle groups you're using. Your body's pretty smart.
You said the word hemoglobin?
Yeah.
That sounds like very familiar. What is that like?
It's the enzyme that you use in your muscles to carry oxygen from your lungs throughout your body. It's what makes blood red.
Oh.
Yeah, it's a big complicated molecule with an ion in the middle of it, and the iron oxidizes, turning into rust, essentially, which you use to carry oxygen around the body.
So then in the case of like animals that live in water, do you have a different cell membrane?
Animals that live in water typically have a lot more myoglobin in there. Did you ever see good
It's
tuna?
like oxygen.
Super dark red. Like how do whales stay underwater for 45 minutes?
No, like this
That kind of stuff.
fish.
They have
breathe water.
they have a lot of myoglobin that they can really attach onto oxygen really, really well. They do a
So
good.
it's the same thing.
There's about a tenth of the oxygen in water as there is in atmosphere. They're very efficient at getting oxygen out of the water, but they also have some physiological modifications for holding on to it more efficiently than we do. Because we don't really need to, because it's like there's oxygen all over the place.
Right.
It's not a big deal. You get out of
So
breath
then
when you walk
what
up the
makes them, like, not
stairs.
survive out of water? If there's still oxygen.
Fish can for a while if you keep the gills moist. So it's like if you ever go fishing and you catch a fish and you wrap it in a wet t-shirt, You can keep that thing alive in there for hours. You know, as
But
long as
without
you keep
any water.
yeah, without it being in water, as long as you keep the gills wet. So they can move the molecules back and forth and do just fine, which is wild. How'd you do today? You did good? You hanging in there? I said this a couple of weeks ago. I don't know if you guys believed it or not, but I'll say it again. You're learning more than you think you are. Right? It's like more is actually getting in than you think is actually getting in. Are you starting to notice that? It's like, you know, you're getting it. It's making sense. It's like, just let the brain do its damn thing. Your task for tonight is to go home and not do biology. Just let it. You've done enough for today. Let it sit. Sleep on it. Let the connections connect themselves. It's going to be awesome. Sound good? Class, you folks are a joy. An absolute joy. You guys are a joy to be with. It's like, I clearly have a good time in here. And I hope you do as well. Goodness me. I have I almost said lovely, but then I wanted to go to delightful, and I didn't know which one to use. Have a lovely weekend. Sleep as much as you can, and eat actual food. Sound good? I love you all. 1, 2, 3. Bye, everybody. Have a good weekend.
Have a nice weekend.
You too. You too. Eat actual food.